All right, so is glucose the only source of energy? No. Free glucose are not common actually in the diet, right? You don't just eat sugar, right? So what do we eat? We eat a lot of different things. All organic compounds can be used in cellular respiration to make ATP. This is an important concept. Glycolysis can occur with, for example, starch. You know that part, right? Which can be broken down into glucose, right? That's easy. And glycogen, which you know is stored in animals. You know that's remember starch is from plants. Glycogen, which is stored in the liver and muscle cells, can be hydrolyzed to glucose too. Okay, now disaccharides also, and proteins. But they'd be digested to the amino acids. Many amino acids can we go on to make other proteins, but the amino acids in excess can turn into glycolysis. So you have too many amino acids. If you eat too much, guess what? If you eat too much protein, it can turn into kind of sugar. And then, but the amino group must be removed. That's called deamination, and that is where you piss out your urea. The urea is the amino. So there it is. So amino acids can go to pyruvate, they can go to acetyl-CoA, they can go to citric acid cycle, but as long as you get rid of the ammonia. Sugars, obviously, we know about that. And remember glycerol? That's the one that becomes glyceraldehyde and into G3P. And remember, G3P was the big one that comes right after fructose, right? And then when fructose splits into two, and then that goes to pyruvate and acetyl-CoA. And remember that. And the fatty acids go straight to acetyl-CoA. That's the most interesting thing. Try to remember that if you can. Fatty acids is the most direct line. Fatty acids straight to acetyl-CoA, glycerol straight to glyceraldehyde 3P. It's kind of interesting to know that. And again, there's another layer, control of cellular respiration. Allosteric enzymes at certain points of the pathway respond to certain inhibitors. Remember phosphofructokinase we had talked about, and I really emphasized it was the guy that added the extra P to the fructose and made a fructose 6 to fructose 1,6, right? There was already a P on here when glucose picked up a P from ATP. Then now fructose adds another P to the ATP. So what's fascinating is how phosphofructokinase controls metabolic pathway. Remember, we had said in your brown fat, if you have too much ATP, you're not using it. And basically, you have too much ATP and it's building up in your system, and you're sleeping all day long, and you're not working all day, but you're making heat with the brown fat. Look at the ATP buildup that comes from all this stuff, right? Mostly this one. Comes back and starts inhibiting phosphofructokinase to actually stop making the next step. So this is a step that regulates either inhibited by the ATP, or in fact even citrates the other way. So you say, oh man, that too much. This is building up too much citrate. Maybe because you don't have enough NADH or something or whatever. I don't know why you would build up a lot of citrate. Citrate would also inhibit this, but here AMP, the opposite, stimulates it, right? So the opposite, this inhibits that stimulate. That makes sense too, because you're like, man, I need more ATP. I'm just sitting around and I need to get it. There's ADP and AMP. AMP, there's monophosphate. That's an important concept there. Okay. This is a good review here. Phosphofructokinase is an allosteric enzyme with receptor sites for inhibitors and activators. Has actually specific inhibitors. Now, this is inhibited by ATP and stimulated by AMP, the monophosphate. Remember, diphosphate and triphosphate, right, is AMP. Now, which derives from ADP. Now, as ATP accumulates, inhibition happens, Phosphorufructokinase is also sensitive to citrate. Citrate accumulates in mitochondria. Some of it passes into the cytosol and inhibits phosphorynthesis mechanism and helps control the rates of glycolysis and citric acid. As citrate accumulates, the glycolysis slows down, and the supply of pyruvate decreases. Now, if citrate consumption increases because of demand for more ATP, glycolysis accelerates and meets the demand, right? Because you're not inhibiting as much, so it's going to do more. So metabolic balance is controlled by these enzymes that build up with these kind of things. The energy that keeps us alive is released, not produced, right? By cellular respiration. Okay, we are, that's good. This is an interesting little concept here. Read this. The fatty acids undergo beta oxidation. This is like considered a major concept here. To 2 carbon fragments and then enter the citric acid cycle of acetyl-CoA. Anabolic pathways can use, don't laugh. Anabolic pathways use small cycles. Remember, they wanted you to remember that. Acetyl-CoA fatty acid. Apparently, it's considered a concept here. So make sure we didn't miss any other concepts. That wasn't even in this. Shows it even here. Oh, there it is. I missed that. Oops. Other fats digest the glycerol, and the glycerol is diverted to glyceraldehyde 3-phosphate. That's glycerol. We know about the glycerol. Remember, I did say fatty acid, but I didn't say how. Ooh. A metabolic cycle is called beta oxidation. It breaks the fatty acids down to 2 carbon fragments and enters acetyl-CoA. And look at this. NADH is even generated during beta oxidation, and they can enter electron transport. So fats make excellent fuels, in large part why? Because they've got the huge CH number of bonds, right? High level energy electrons present and the CH bonds that share equally between C and H compared to those of carbohydrates. So a gram of fat is interesting, produces twice as much ATP as a gram of carbo. Unfortunately, this also means that people trying to lose weight have to really work hard, because to lose a gram of fat, to work twice as hard, right? So we talk about biosynthesis or phosphorufructokinase. Remember that. Just real quick, where was phosphorufructokinase? This guy, remember, it was a hexyl kinase added on a P, and then he flipped it to a fructose from the glucose, and because when the P was added, the P stayed, and now we add on another P. This is the regulated enzyme that turns it into, and now it's with the addition of a P. So phosphorufructokinase takes a P from an ATP, picks a P from an ATP, and that adds it to the bisphosphate, right? And then and that's the glyceraldehyde. Remember, that came to glycerol, right? Because it almost looks like a glycerol kind of thing. But it adds on a P, and then that P is already there. And then remember, glycerol, this is the guy that actually gets into the dehydrogenase, right? The triose phosphate. Dehydrogenase is the guy that actually now, remember, makes NADH, right? And he's the one that strips 2 electrons and 2 H's, right? 2 electrons per NADH, right? And that's why you had 4 electrons per NAD. Remember, don't forget that. Remember that 1 NAD always gets 2 electrons. Okay, now, just a quick reminder review, and then these are little pathways. You don't have to remember those things, but you just need to know this one, that we remove the ATP right before when pyruvate, which is that interesting, funky-looking double C and double L. Kind of looks like a funky guy there. Okay, all right. So I think we beat up respiration pretty good. I think we covered it as much as I can think of. I can't come up with anything else. We beat this thing up pretty good. Quick reminders, resummaries. Inputs: 2 oxaloacetates combined with 2 acetyl-CoAs to make your citric acid. Okay, I don't know why they added this little tidbit in there. So obviously, they want the little detail. They want you as a concept to remember. And in the end, they make 6 carbon dioxides, 8 NADHs, 2 ATPs, and 2 FADs. So the concept is this, is that carbon dioxide is made in citric acid. Carbon dioxide is also made, where was one other step where carbon dioxide was made? Remember, pyruvate made carbon dioxide when it turned into acetyl-CoA. So that was the pyruvate dehydrogenase, remember? And it added the acetyl to the pyruvate, and remember, it took on an NADH. Remember that part, okay? Just to review. Do you want to do these real quick questions? I can get some questions for you. We can do those, but you can crank these out now. Most CO2 is released during one, glycolysis, citric acid, lactate ferment, or electron transfer. We know citric acid, remember, it generates tons of carbon dioxide, right? We know that part. We should know all this stuff now. What is the oxidizing agent in the following reaction? The pyruvate gets oxidized. Always, the organic molecule always gets oxidized, right? So this is interesting. It's fascinating. We're here. The oxidizing agent actually is tricky, this one. This is the tricky one. Pyruvate is actually, hold on a second, that's a tricky question. That's number 5. Hold on, this is page 186. Hang on. It's the opposite of what you think, because that's fermentation. So, chapter 9, hold on. Hang on. I didn't see that question. That's a good one. Come on, I'm glad we looked at that. Chapter 9. Hi, Mama. Chapter 9. Where are you? Chapter 9. No. Is this chapter 9? Yeah, chapter 9, yeah. Questions, testing your understanding. Question 5 is D. Pyruvate is actually the oxidizing agent. This is weird. So normally, remember, the reason pyruvate is the oxidizing agent, remember this one. This is one's tricky. As we had talked about, pyruvate, in this circumstance, is removing the electrons from NADH. Normally, NADH is delivering the electrons to oxygen. In this sense, pyruvate is actually taking away the NADH electrons from here to make this NAD+. Right? This is the first time when NADH is actually giving it up to an organic molecule. In this circumstance, it's giving it. Normally, NADH is delivering to an electron transport, right? So in this circumstance, this is sort of interesting recognition of a point that helps you understand a little bit of summary ideas. Remember, 34% energy is glucose, is ATP, the rest is heat. ATP synthase, interstellar membrane, down here, it takes 4 of these, right, to go down to make 1 ATP. And we know about this. So these guys, FAD starts a little bit late. Okay, that was a good question. When electron flows from the electron transport chain, which of the following changes occur? The pH of the matrix increases, doesn't it? The pH increases. The pH increases the matrix. Why? The pH increases the matrix because you're pulling out the H from the matrix. So the pH goes up in the matrix. It becomes more basic because this becomes more acidic. So the pH goes down in intercellular membrane space, and so you get a pH gradient, right? So that's a good question, right? So that's a good question. That's an important thing. So the electrons create differential, right? And that's just important. That is definitely the answer. The pH increases. B, ATP synthase pumps protons by active transport. the electrons gain free energy. No, they don't gain free energy. The electrons lose energy. NAD plus is not oxidized, it's reduced, right? So you're adding electrons to it. So that's not true. And ADP synthase pumps protons by active transport. No. What is the process of how ATP synthase works? Here is the main concept. Is ion pumps usually use ATP as an energy, ion pumps. So it's like an ion pump. ATP is used as energy. Oh, so normally active transports use ATP as an energy. This is what's different. We're not using ATP as an energy. What are we using? Uses the energy of the existing ion gradient to power the ATP synthesis, right? So the ATP synthesis occurs because of the gradient to generate ATP. That's where the power is coming from. So the power source for ATP is the difference in the concentration of the EH, the pH difference that we had talked about. That pH difference is what drives this little enzyme. This important resummarization on the opposite side. This will be on your test. I forgot to mention this also, this little summary. So it's good to review some important concepts. I'm glad we did this little section, these questions. Let's do another one, number 4. In mitochondria, exergonic redox reactions are the source of energy driving prokaryotic ATP synthesis, provide the energy that establishes the proton gradient. Provides the energy okay. Reduces carbon atoms are coupled via phospholipid intermediates to endogenous processes, are the source driving ATP synthesis. Now, prokaryotes, that's very tricky, right? Do prokaryotes have mitochondria? I don't think so. Remember, they have their things the eukaryotic. So prokaryotic, they're on the plasma membrane. Remember, their electron transport chain was on the plasma membrane. They see this. They try to mess you up here. Mitochondria provide the energy that establishes the proton gradient. I mean, the exergonic redox reactions. So the exergonic redox reactions does provide the energy that supplies the proton gradient, right? Because the gradient is created by that electron from the NADH that was created, right? So that is really the driving point, right? So that's really the most important point. I don't know. And the final electron acceptor of the electron transport chain in aerobic oxygen is always oxygen, right? Remember, the oxygen pulls everybody downstream. Make connections. The enzyme phosphofructokinase is allosterically regulated by ATP. Considering the overall glycolysis, would you expect ATP inhibit or stimulate this enzyme? Oh, we know that. ATP inhibits that enzyme. That's easy. Okay, the proton pump inhibitor shows, okay, the proton ATP is like this. Compare the process of 3 figures, active or passive transport. Well, this is what we already talked about, that part. We just did. Okay, real quick. Interpret that phosphofructokinase is an enzyme, early step breakdown. So they're really hammering in this concept. Jesus. Considering the graph under which the fashion, given glycolysis regulation, explain the mechanism by which factor fructokinase differs between ATP. Explain why this sense makes sense and why this way. Okay, anyways, why this makes sense. If you have a low ATP concentration, phosphofructase activity level is going to really increase. And phospho-6 concentration is going to go down, right? Because you're going to have less of this because you're using it all up. You're using it all up, the phosphofructokinase, as that becomes more active. If you have high ATP concentration, you have too much of that, this is going to build up. right? This will build up real quick. Okay, so this is the back substrate, right? So this will build up if you have too much ATP, because this is not using the original substrate, right? That's basically what they're trying to say in this. They're making you think about it again. Again, the pH difference, again, they're really driving this concept. pH difference between the membranes, okay? So we talked about that, talked about the pH difference in this. So really they're hammering it home on these question marks here. We talked about this thing here. They really love these questions. And then we talked about this. This is a good summary. I'm glad we did the last set of questions. This is an important little 20 minutes, kiddo. And now we're going to get a fast silence. Bye.